Welcome to today's virtual seminar, Modeling Resource Capture in Plants, presented by Dr. Colin Campbell of Decagon Devices.
Thanks, Rhea. Welcome to the workshop that we're talking about today on modeling resource capture in plants. My name is Colin Campbell. Just to give you a little bit of background about me, I have a PhD in soil physics from Texas A&M University. What I did there was actually study resource capture in irrigated rice crops, and we did some field scale modeling and monitoring of carbon dioxide uptake in that crop. And today we're actually going to use some of those data. I actually pulled them out for this workshop, for this seminar, to actually look into those data. They're a little bit old now, but they're really pertinent to this topic, and I hope you enjoy looking at those data and learning a little bit about this resource capture in crops. Right now I'm a research scientist here at Decagon, and I'm also part of an adjunct faculty member of the Department of Crop and Soil Sciences at Washington State University. Long ago, I used to go out to the potato fields of central Washington with my father. I used to love those trips. We'd go out there and Dad would do whatever he did. I didn't really know back in those days. And he'd dig up some potatoes there and feed us potatoes out in the field. And it was only as I grew up and started to go to school and study this kind of stuff that I learned what he was trying to do. Dad was actually studying with Dr. Bob Kunkel at the university how to improve potato yields here in the Northwest. And that Dr. Kunkel over the years has really done great service to this industry. But here I'm showing some plot studies that Dr. Kunkel did, where they were looking at the potential yield or the predicted yield of these potato plots versus the actual measured yield. And this graph shows a very interesting thing, that there are a few of those plots, some of the years, that were able to approach the maximum possible yield of that potato plot, but most of the potatoes really didn't ever come close to that optimal line. And the very simple question that Dr. Kunkel asked my father at that time was, well, how can we actually get close to that line? How can I achieve the maximum possible yield in my potatoes each time? Well, that really brings us back to the question that we're all here to try to answer, which is, how can I predict and understand resource capture in my crop or in my plant, in the plants that I'm working with? And that has to do with some of the things that we're showing now. On the bottom, the line that I actually showed in the last graph is predicted by this equation, which says that the yield of a crop is going to be just the harvest index multiplied by the biomass and total crop biomass of that crop. And that's that one-to-one line on that graph I just showed. Now, how do we find this biomass? Well, that's actually relatively simple. Biomass is the radiation use efficiency of the crop multiplied by the fraction of interception of that crop relating to the canopy structure. So the radiation use efficiency, that depends on biochemistry of the crop. We've got our fraction of interception that depends on the canopy structure, and then the intercepted, the actual total solar radiation coming into the crop. So we can conclude from the relationships that I just showed that some of the plots yield at the maximum possible level for that environment and the growing conditions. That was that line there and those dots that approached the line. But all of the others that didn't get there, the less yielding plots, they had other issues with them. For example, one of the reasons they didn't yield high enough was that they had a low radiation use efficiency. Why would they have that? Well, there are a lot of things when we say radiation use efficiency that might cause that. Things like disease, stress, and other things that lower that value. And they really are environmental concerns, and essentially falling off that line related to the environment the crop was grown in. There are other things that could have a factor as well. This fraction of interception. You can imagine a plant canopy where, in fact, the leaves are not growing to the relative size that they could because of some problems, for example, with water stress. So that we might have a very limited canopy where they're not intercepting the radiation in the way that they would typically do. Or we could have a senescing canopy, something that we'll actually talk about in the rice data, where that canopy begins to senesce and we actually don't have the interception of radiation like we would in an optimal growing canopy. So when we think about how can we create higher biomass and eventually higher yield, we saw that relationship with the harvest index just a moment ago. And we ought to think about, well, where does this come from? Well, Dr. Kunkel could improve back when he was doing this, his potato yields by finding varieties of potatoes that would have a higher radiation use efficiency, and we'll talk about tweaks to that a little bit later in the seminar. And we could also lengthen the growing season, something a little harder to do without picking up and going somewhere else. So for this talk, we're going to go through a discussion where we first talk about the resource capture in plants and learning a little bit about resources and capture in those terms. Then we're going to talk about how we can actually field measure biomass accumulation and biomass production and relating that to light capture. After that, we're going to talk about radiation use efficiency and predicting biomass from that. We're going to talk about how to measure that, because it's critical to really think about how to measure these things. We can guess at it, we can model it and all those things, but how are we ever going to know that what we've done is right? Well, we actually have to go out to the field and measure it. So that's the next step. Talking about intercepted photosynthetically active radiation, being able to measure that. Then we're going to talk about remote spectral indices, just some fun stuff that we've been working on here, and I think we'll see more and more use out of here in the future. And finally, we're actually going to talk about conditions where the radiation use efficiency model that I just showed you may not be as useful. And we're going to talk about water use efficiency, where we can link the water use efficiency to carbon dioxide uptake and photosynthesis, and have another model that we can use where, for example, conditions aren't well watered. And when we think about that, this work that Dr. Kunkel did so many years ago, that was all in irrigated agriculture, where he was irrigating those potatoes to optimum levels. What if that's not the case? How do we produce a model where we can actually discuss a situation where that's not happening? We're going to touch on that a little bit at the end of the seminar as well. Before we go on, we've got to actually define the term resource and the term capture. And this was done by Dr. John Monteith several years ago in a paper in 1994, where he said a resource is a form of energy or of matter that plants need in order to grow or to reproduce. He defined the capture as the process by which any organism above or below ground removes a resource from its environment to maintain its metabolism. Those are the things that we're going to talk about today. First, about resource and about the ability to capture that resource by the plant. Now, we're going to focus in on three resources, three things that I would like to study, being an environmental biophysicist and someone who looks at the soil-plant-atmosphere continuum and tries to understand this resource capture, how it happens, and how we can eventually go from making measurements in the field to actually modeling this. And we're going to talk about these three things. First of all, carbon dioxide, and light or solar radiation, and then water. Now, you're seeing there at the bottom of the page, we're graying out essential nutrients. Well, sorry, we're not going to talk about that today. We're going to acknowledge that creates the problem, and we're going to sometimes fall well off that optimum line because we're not touching on that, or because we don't have those nutrients, but we're not going to talk about it today. We'll leave that for another day. Let's talk about some basic principles of biomass accumulation. These are all things that we know fundamentally, but we've got to create a foundation for this discussion so that we can move on and actually identify the things that we're interested in. So plants, of course, they take up CO2 to produce biomass, and we know this. And the cool thing is that we can actually measure that CO2 in the process and actually fine-tune our understanding about the biomass uptake. And we'll talk about that in a minute. We also know that plants require water for biochemical reactions. And they get this water, or they have this water inside the plant, and when they need CO2, they exchange that water for CO2. Now, I was in a seminar just a few weeks back, and they said, okay, that's not the only reason that the plants exchange water. They do it to cool. And of course, that's true. That's not the only reason they do that. But to maintain a hydrated state for biochemical reactions, plants must regulate their water vapor loss and at the same time take up CO2 for those same reactions for photosynthesis to take place. We also know, and this is going to help us in being able to learn more about plants and about these interactions that we're talking about, plants exhibit different absorbance or reflectances, and these ratios at certain wavelengths of light. And they can be associated with things like light capture, photosynthesis, and water stress. And we'll talk about at least the first one, this light capture, and we'll probably leave those others for another seminar. But those are all interesting topics and areas of a lot of work right now. So why are you here? It would be great if I could actually get you to raise your hand in the audience and tell me a little bit about the research you're doing, because I could tailor this to what you're doing. But I'm going to make an assumption here that all of you, or most of you anyway, are actually here listening to this seminar because you want more information on maybe three areas. First of all, ecosystem source-sink relationships for CO2 uptake. That's what I studied during my PhD. That's where I enjoy reading more about that today, and it really is an area where there's a lot of interest right now, trying to understand the sinks for carbon dioxide, greenhouse gases, where they're going and what they're doing. Now, others of you may be here to learn more about plant growth and biomass accumulation to try to better fine-tune your models. Maybe you're doing remote sensing. Maybe you're trying to do some ground truthing and trying to match some of those up, looking at plant growth and biomass accumulation. And some of you, much like Dr. Kunkel, who I was talking about earlier, are here to look at yield and being able to predict yield and using some models to predict yield and trying to build on some of this understanding to do a better job. And we all know that at the very basis, that this improving of yield right now is critical for our growing planet. Let's talk about some ways to actually measure biomass production in the field and talk about its association with light capture. And here's where I had a little fun, because I went back and mined a little bit of my old work in this area, and I've been around the world doing these seminars in different places, and I've got to see some interesting applications. On the top left-hand side of the screen, that's my old student worker actually harvesting a square meter of biomass. So that the very lowest level, if we want to understand biomass uptake, we can do as I did and as Cody did. We went out, took a square meter of a PVC, closed our eyes and tossed it out there in our rice field. This was a commercial rice field. We had a lot of area to work with. We just tossed it out there 6 or 7 times during the growing season. And just went out there and sampled everything inside that 1 meter square at a few different locations within our measurement area. Once we collected all that, we'd take it back to the lab. We'd allow it to dry. We'd weigh it, we'd weigh it, and then it'd allow it to dry. Weigh it again, and we get this dry matter of the plants. That way, we actually were sampling directly the biomass. Another way that you can actually do this, look at this resource capture, this biomass accumulation, is in chambers. And on the top right-hand side of your screen here, you actually see some chambers down near Sydney, Australia, that I visited a couple of years ago, where they were actually containing the entire tree that they were studying. Inside these chambers, they could control the conditions of this tree, and they could look at biomass uptake, among a lot of other things. This is a really cool area that they were doing this in. And finally, down in the bottom middle, this is an eddy flux system. And we can measure the carbon dioxide coming into and going out of a system by simply measuring the eddies that move past a three-dimensional sonic anemometer and sampling out of those eddies the concentration of carbon dioxide and other scalars of interest like water vapor. These are really good ways to look at the biomass directly, and they're great for calibrating our models. But you can see from the drawbacks list there that a lot of people don't have the money, aren't invested enough, don't have the time to go to these systems. So really have to look at the models to be able to do this. Well, let's first be sure that the relationships we're going to talk about are solid enough that we can be confident in them. We're first going to talk about the very foundational understanding of why we might do this. And I showed you this equation earlier where I said biomass is related to the fraction of interception times the radiation that's coming in, the solar radiation times this radiation use efficiency. And this graph is just a classic graph I like to use in my class from Dr. John Monteith back in 1977, so it's pretty old. But it shows a linear relationship between an intercepted solar radiation that accumulates over time and the total dry matter that's produced by these plants. That's accumulated over time, obviously. Now, any two variables that accumulate over time, or that accumulate on their own, probably have a linear relationship. So that doesn't necessarily mean that there is a causational relationship between any two variables. But here we know that since plants are using the solar radiation for photosynthesis, and whatever is intercepted by that plant is going to be correlated somehow to total biomass uptake. Shouldn't surprise us that we have a really nice linear correlation between all these different crops. But notice here in each of those lines that they're a little bit different. We have apples, barley, potato, sugar beets. The slope of each one of those lines is a little bit different. And that's the slope of that line is the radiation use efficiency. So tuck that back in the back of your mind. We're going to talk about that in a few minutes, but that's what that is, and it's a little different for each crop. Now, does it differ through the season? Well, I'll let you be the judge when we get to a little bit of rice data here in a moment. So that picture I showed you, that eddy sampling system, or eddy flux system, was actually the system we used out in this irrigated rice field in south central Texas about 12 years ago. We had a lot of fun doing this because in this field we had a large flat field, we had water covering. This was an irrigated, flooded situation, so that we can actually get at a lot of the interesting parameters in terms of CO2 flux and get directly at photosynthesis. But there you're just seeing the flux tower there with data acquisition control system. There's a lot of stuff that went into that we don't have time to talk about today, but we did collect a pretty good data set. And this is showing that data set. The CER, I didn't actually change this graph. It's talked about a little bit differently. Net ecosystem exchange, sometimes it's called now, but we called it carbon dioxide exchange rate, or CER. And I'm going to show several graphs that actually have CER, or carbon dioxide exchange rate, on the y-axis compared to something on the x. And here we just actually have a really nice graph showing the total carbon dioxide exchange rate summed over a day across the entire season. Now it's interesting to know that, and I didn't really know this before I started working with rice, that rice actually has a primary crop and a ratoon crop, and I didn't even know what ratoon was. Ratoon is basically the second crop. Rice is a tillering plant, and it'll grow again after harvest and tiller up. It won't be obviously as strong and robust a plant, but you can harvest a second crop off the rice. Now if you look at the carbon dioxide exchange rate, you can see a lot of the phenological changes in that rice plant over the primary and ratoon crop, and a couple I've actually labeled on the grass. You can see the panicle differentiation, meaning the beginning of the fruiting structure. Then you can see the actual heading of the fruiting structure, or anthesis, there marked. And the time between panicle differentiation and heading was actually the maximum carbon dioxide uptake during this time, which it shouldn't surprise us. This is where the plant is putting on the most biomass there. See where the crop is drained there. It hurt us a little bit when it's drained. We actually have an addition of CO2 that's actually moving out of the soil, because once it gets drained, we have the situation changed from anaerobic conditions under the water to aerobic conditions. So that's just a little bit of information about what we saw out of the rice crop. But how does that tell us about biomass? Well, let's actually start looking at some of the data we collected. First of all, really we wanted proof that if we're going to make these measurements on a field scale, we want to be able to match them up with the only real absolute measurement that we can do out there, which is that 1 meter square sampling. So you can see all the samples we took over the first season and in the second season. Those are both for primary crops. First on the first year, we can see that the data match really well. Now you say, hey, wait a second, did you just throw that 0.77 out there because that fit the data well? What we did was actually take some of that dry biomass that we collect in the field, we pulverized it, and then we did a total carbon analysis on it and calculated from the chemistry the actual relationship between carbon dioxide and the biomass. So that's not a pie in the sky number. That was actually calculated from the total carbon in the rice plant that we had out in the field. And as these graphs shows, we had great correlation there. What's the point of these graphs? Well, it's not the end of this discussion. It's just the beginning. It's to say, look, we can actually measure carbon dioxide uptake on a pretty fine-tuned basis. And by the way, it does match with the field biomass we've sampled. So these are good data. One of the things we really wanted to know at the outset was, well, as these plants change, what happens when we actually look at carbon dioxide exchange on a daily basis per unit leaf area? So how is that changing over the season? It was interesting to actually look at this and find out that in fact we had a gradual downward trend over the season as we went through panicle differentiation, went through anthesis, or that heading, that in fact we just had a gradual decrease in that. You can see the leaf area index superimposed in the black dots. Those are our measurements. We actually fitted it with a function. That's in green. And you can see how that changed over the season. And in fact, it really dropped off after anthesis as we've got a significant leaf senescence. Now just keep that in the back of your mind and to look at the next graph, because you can see some of what was challenging to us once that came off. So what we wanted to do here was look at radiation use efficiency, because we wanted to know how radiation use efficiency changed over the season. That way we can use a model to predict the biomass uptake. So here we are. We've proved that the biomass uptake can be accurately, at least from what we showed there in those graphs, can be consistent with actually the field biomass and the carbon dioxide exchange rate can be tied together quite closely. Now let's look at the carbon dioxide exchange rate. Now each one of these points that you see there is now not a day, but we're talking about a half-hourly average. So we split them up by leaf area index, and what we wanted to do was look at the slope of that line, the slope of the line between the carbon dioxide exchange rate and the intercepted photosynthetically active radiation. That slope there is our radiation use efficiency. And what we wanted to know was, are these lines statistically similar or different? What we found was, up to this post-anthesis point where you really saw that drop off, that in fact our radiation use efficiency was very similar. Those slopes were not statistically different. So we found that we could use, essentially for the bulk of the growth of this crop, the same radiation use efficiency value to actually predict biomass. Now, we also learned something interesting. It's just a side note here that rice reaches light saturation at high intercepted photosynthetically active radiation. Now, I only point this out because a little later in the presentation I'm actually going to say, hey, wait a second, this radiation use efficiency is actually high when the diffuse radiation is high, and that really is true, and that essentially just brings the radiation off that peak curve where it's reached saturation and down onto that linear relationship there. So just keep that back in mind for a comment I'll make later. So now we've actually talked about how to field measure biomass and the fact that our biomass sampling does match up with other things that we can do out there, like CO2 flux measurements, and that our radiation use efficiency is fairly consistent across the season. And we could see places where it's not, but generally we can use the same RUE value, radiation use efficiency value. Now, let's go on and talk about how to use this radiation use efficiency to predict biomass accumulation and yield. Here's that equation again that I started with earlier that just said the biomass accumulation is equal to the fraction of interception times the radiation use efficiency times the solar radiation, the light coming in. Now, this assumes, and I mentioned this earlier, that we have a situation where we're well watered. And notice that I used a very convenient data set, which was one of the ultimate crops in a well-watered situation is actually flood irrigated rice. We also didn't have any nutrient deficiencies out there that I could spot, and we did collect all the information from the farmer that was taking care of the field, who actually showed us all the nitrogen that had been put on, a little in excess of the recommendations for that area. The interesting thing is that radiation use efficiency number that I showed was a conservative value from the 2 years that we looked at it. For that variety of rice, we had a consistent radiation use efficiency. Now, I have that little comment on the bottom. I already pointed that out, that sometimes two accumulating variables can be linearly correlated. But we already know, and I've told you that that's not a big deal for us because we know this is not a spurious correlation. Claudio Stockel and Armin Kamenian, 2009, put together a list of several radiation use efficiencies over ranges. They've spent some time modeling crops, and here they've put together some information that may be useful to you if you don't have values for your own crop. And this is pretty easy. If you were trying to do some modeling, you just needed some numbers to get at. You've got your C3 plants, your C4 plants, and some of the other things like oils, legumes, and tuber and root plants. Some things that you see from this table are that C3 plants have a lower radiation use efficiency than C4 plants. And we'll talk about that in a little while. That's because of the intercellular space CO2 that they're able to take up into the leaf and be able to have a lower concentration inside there that will actually be used in photosynthesis. Legumes tend to have lower radiation use efficiencies as well, but tubers, high sink strength things, or plants, have a rather high radiation use efficiency. Some other things that you ought to be thinking about are the fact that radiation use efficiency decreases with increasing vapor deficit. And I said why that might be, because we are exchanging CO2 for water vapor. If there's a bigger sink for water vapor, we can see that radiation use efficiency may be pulled down. And I already mentioned that. Remember a couple of slides ago, radiation use efficiency increases with increasing or strong diffuse radiation. And we actually found that out in the rice when in the second year we had these large fires down in Mexico that sent these large, very high altitude clouds up over our rice field. And during that time we calculated the radiation use efficiency. And although it washed out in the general data, we could see that it was higher for those specific times. So at this point, we need to talk about how do we measure radiation use efficiency and light interception. How do we make these key steps to be able to grab the direct information for our crop or the plants that we're working with? And we can do these one of two ways. We can either measure the intercepted photosynthetic radiation, or we can measure the remote spectral indices. And I mentioned those two before, but let's actually get into those measurements. So here's our equation that we're talking about, the radiation use efficiency. And we have the components. We've just rearranged that equation I showed you a little bit earlier. So now we have the radiation use efficiency is equal to the biomass production divided by the fractional interception times the solar radiation, either measured using total solar radiation or photosynthetically active radiation. Maybe you can do either, but you've got to use that in your model eventually, so you have to be consistent. Now, when you look at that, I have arrows here in terms of how you might be able to measure these things. Of course, in most cases where we're trying to actually do this for an individual crop or something like that, we'd measure biomass accumulation just by going and actually frequently sampling out in the field. To measure the solar radiation, that would be fairly straightforward. We just need a pyranometer or a PAR sensor to set up and measure over time. But the fraction interception, that ends up being a bit more of a challenge. We're going to actually have to go out there and make repeated measurements in the canopy to get that fractional interception. Now, of course, there are cases where this is not true. I like this corn canopy. In fact, the rice was very similar to this. You noticed our leaf area index going above 6 in the rice. And basically, at that point, there was very little radiation getting down to the crop surface. What that means is a leaf area index of 6 means that there are 6 meters squared of leaf area per 1 meter squared of ground area. Essentially, we had leaves stacked on top of each other 6 layers thick. And you can imagine that that is not going to let a lot of radiation to the ground. Fi would be very close to 1, just like it is here in this corn canopy. But you can imagine when that corn was just starting to grow, as we're seeing in this picture on the right-hand side, that's not anywhere close to being a complete canopy cover. And there we're going to have to go measure it. Even in the rice, that's actually the picture on your left-hand side there, that rice is also still not quite a full canopy. Although it's getting close, that leaf area index was somewhere around 2.5 to 3. And we need to measure both of those situations. So how do we do it? Well, the first way to do it is with a septometer, an instrument that measures interception in plant canopies. Now, light below these canopies you can imagine to be fairly variable. And you've got to actually go out and make a lot of these measurements to be able to get an accurate value of this light interception. This instrument you're seeing here, the LP80, actually has 80 little light sensors along this bar, an 80 centimeter bar, that actually can make rapid measurements of light interception. So how's that done? Well, we either use an external sensor, a radiation sensor above the canopy, or just put that sensor, the LP80 above the canopy, take an incident radiation measurement, and then take many measurements leveled below the canopy in several locations to take the below canopy measurement. And the LP80 actually computes this TR value, or I often call it tau, this transmission coefficient below the canopy. Well, that's the radiation that is actually making it through the leaves to the ground. We still got to do a 1 minus that tau value to get this fraction of interception value. And also, by the way, we've got to be able to convert it to a daily value. And if you go to the user's manual of this particular instrument, you'll be able to see how to convert up from something you measured at some point in the day all the way to a daily value. And then you'll be able to get this Fi value, and you'll have that. That's not the only way to do it. And sometimes you want to make these measurements over time. That method I just showed you with the the septometer to measure interception is probably the most accurate way, besides simply going out and biomass sampling and looking at the leaf area there. But another interesting way that we've been playing around with, this has been around quite a long time, is this NDVI, or Normalized Difference Vegetation Index, which compares light at two different wavelengths at 800 nanometers and at 675 nanometers. It compares the reflection off the surface of whatever crop or canopy that you're looking at, and it takes the difference between them, divides them by the sum of them, and that NDVI value then is correlated with the leaf area index or the intercepted value. In the literature, there's some discussion on that. We can take it to be approximately equal to the intercepted radiation in that crop. And there's a paper available on that I've noted down there at the bottom of the page. But for you, by the way, we've got all these papers, all these references at the end of the presentation today. So if you're interested in looking up these papers and they might help, just hold your horses. You don't have to write these down quickly. We'll get to it in the end, or you can download it from our website. Now, what about this canopy? This is a grass. There's just not much there. You're certainly not going to get a septometer underneath this canopy, and it's unlikely that an NDVI sensor would give you any good information here. One of the ways that we've worked with this before is actually to take a digital image of this surface, put it into something like a program that will actually analyze, Photoshop for example, analyze the green versus brown pixels, and then do some analysis of the canopy cover, and you can actually get it from there. There are also programs out there that you can purchase that will do this analysis for you. So you can collect your canopy cover in that method too when you have very sparse canopies. So all this discussion of radiation use efficiency was fine when we had a well-watered crop and there were no nutrient deficiencies, but that doesn't happen all the time. Are there other methods for modeling this kind of stuff? Well, there is. And we can do that through water use efficiency as a predictor of photosynthesis and biomass accumulation. So let's look at some of the equations that we can work with that. And now first, what are we talking about? We know that plants take up CO2 at the expense of water vapor. I talked about that right at the beginning. And here's a nice little diagram from a plant physiology book by Tays and Zeiger that just shows the CO2 moving into the leaf and the water vapor moving out. And those little squiggly lines in there represents resistances or conductances to water vapor as they move through those stomates. Now, it's probably not something that comes as a surprise that water vapor and CO2 move in concert with each other. It's not like in that diagram where one goes out and one comes in. They're going together. And so to analyze this, we can start to put some of these equations together. First of all, we can talk about how to calculate photosynthesis. Photosynthesis is just the conductance to carbon dioxide gas times the concentration difference of carbon dioxide outside the leaf and inside the leaf. We can calculate transpiration in the same manner, where we say the conductance to water vapor multiplied by the concentration difference in water vapor. Or we can move that a little forward, the conductance to water vapor times the vapor deficit. That will give us our transpiration. And since we know that they actually move in concert, we can combine those together to give us what we call a transpiration efficiency. And if we work through the math on that, that eventually gets down to this K value, which is our normalized efficiency, divided by our vapor deficit. Now, this normalized efficiency value, why do we actually go and use that value? Well, the K value is kind of useful. It's very nearly a constant. The difference between the conductance to CO2 and to water vapor is just a constant, 0.7, that ratio. And that ends up being just from the conductance of those two molecules in air. Now, the concentration difference in CO2, well, that's not quite a fixed value in terms of the concentration in air, but we can measure that. We know approximately what that is. We also, for different types of plants, know what that concentration is inside the leaf. So we actually that's very close to a constant. We can calculate it fairly easily. It's not something that's changing over the hours of a day, for example. Now we can come up with actually a rather simple water capture model that says that the photosynthesis is related to this normalized efficiency times the transpiration times this vapor deficit that we talked about. So now let's actually jump in and talk about some of the things that we learned from this. So the first thing that we know from this simplistic model is this ratio. The P over T is called that transpiration efficiency, or the water use efficiency. Many of you, I think, are much more used to calling this the water use efficiency value, which I'll use here in a minute as we describe some things in rice. It's a mass of carbon fixed per unit water used. And it's interesting to note here that it's anywhere between 0.1 and 1%, that value, or 100 to 1,000 times the water use versus the carbon dioxide fixed. So that's quite an immense ratio there. We also know that photosynthesis is proportional, and dry matter production is proportional to transpiration. So if somebody came up to you and said, well, I'd like to actually engineer a plant to basically use next to no water, but put on biomass, you could quickly tell them, okay, physics is actually limiting this process. There's no way to produce biomass through photosynthesis without actually transpiring water out of the plant. Now, transpiration efficiency, of course, that really depends on the environment because it's got that D term in the bottom, our vapor pressure deficit. And depending on our environment, that's going to change. Arid regions, for example, they're going to have a high vapor deficit and a lower dry matter production per unit water use, meaning lower water use efficiency compared to humid regions. And as you might imagine, transpirational efficiency varies dramatically over a day and even over a season as vapor deficits change. So in this case, this model, whereas the other model was a little more robust, the radiation use efficiency over a season, this model actually requires more continuous input. Well, I wanted to throw up a little graph to show you why this is. You saw in that radiation use efficiency that our data, although scattered, and you'd expect that because we're on a flux tower measuring this, in this case, our water use efficiency information, that's still there, but notice our carbon dioxide exchange rate, half-hourly basis again, versus our evaporation is much more scattered in these graphs, and we have much more of a changing relationship there. So we're going to have to actually make these measurements on a more consistent basis. You can see that they change through the day, and they're not nearly as consistent. Now you might say, now wait a second, you say evaporation down there, we're talking transpiration just a second ago. Yeah, in this rice crop, we didn't actually, well, I did partition it, not in this particular slide, but I did partition it so you can talk about that. But without partitioning, we don't know what that scatter is from, basically. Now, what else can we conclude from this? Now, transpirational efficiency depends on that concentration difference between ambient CO2 and internal CO2. Okay, that 0.7 isn't going to change. That ratio of conductances is going to be the same, but that CO2 will be different. And you're immediately jumping to the fact that we talked about earlier and something you already know, which is species that maintain a low concentration of CO2 inside their leaves have a higher transpirational efficiency. And that leads us to a discussion of, for example, C4 plants, which have a very low internal CO2 concentration compared to C3, so C4 plants have a higher transpirational efficiency. And obviously, one consequence of our increasing atmospheric CO2 is that we are going to increase transpirational efficiency in these plants. Here are some of those normalized efficiency values, those K values for several crops here. You got corn, potatoes, alfalfa and soybeans. These are a bit old. These you can kind of stick into the model just as a way to try things and maybe match some of the things you're doing. But like I said before, this has to be, there's a lot more measurement that's got to go on. And so we really need to move on and talk about how we actually grab that transpirational efficiency from your crop. So one of the things we've got to do is measure our biomass. And we measure that biomass. We already talked about it through typically destructive sampling, harvesting, weigh dry, weigh method, and look at the end weight of the biomass. Now, we can measure the transpiration by doing some kind of container method. That's, you can see on your right is a picture from a scientist named John Widtsoe, who actually did these measurements clear at the beginning of the last century. So this has been done a long time. But the problem with actually monitoring the weight over time, and with that kind of system, is in fact you're getting both evaporation and transpiration. How do we actually split those things? And I did mention that partition just a little earlier. Well, we can do that, but it really goes back to a question of this well-watered crop, or doing some math, doing some analysis beyond this, because what we could do to partition this would be to use a model like the Penman-Monteith to get our evapotranspiration, our potential evapotranspiration, meaning the evapotranspiration in a well-watered crop, then split those two things, where our potential transpiration is the fraction of interception times the PET, potential evapotranspiration, and our potential evaporation is just 1 minus the fractional interception times the PET value. Now, to even start at this analysis partitioning, we've got to have the fraction of interception measured to be able to actually partition those. But once we have those down, we can compute that transpiration value and get out that transpiration efficiency. Now, how to determine this vapor deficit? Well, vapor deficit is actually fairly easy to measure. As you know, all we need to know is the saturation vapor pressure, which takes a measurement of the air temperature, that's accurate, and a vapor pressure measurement, which we need the relative humidity and the temperature at which that relative humidity was made. Then we can put that together to get the vapor deficit. And that's one of the most commonly measured values out in research. Now, one of the things you got to notice is when you're putting together this model, it's usually tempting, like I have done on occasion when I'm looking at some of the parameters in the field, just to grab a daily average. But if you grab the daily average, you might be in trouble a little bit. So you notice from this figure, in fact, if we average the vapor deficit across a day, we get a value of 1.2 kPa. But if we actually took the daytime average, where we're actually getting the photosynthesis occur, where we've got that exchange between the air and the leaf actually going to photosynthesis and the CO2 coming in, with the CO2 coming in, we get a value of 1.2 kPa. So just be aware that you've got to make some adjustments to those values. Just can't naturally take the average of the entire day's vapor deficit. So how could we measure this transpiration efficiency more directly? Well, we already talked about the flux tower, and another way of doing that would be the Bowen ratio system. Other people have done it through carbon isotope discrimination. We can measure simultaneous transpiration and photosynthesis with an instrument. We can measure stomatal conductance, and we can measure, it's been shown in literature, can measure the canopy temperature. Now, a lot of these things we're not going to have time to get into, but we can talk briefly about measuring stomatal conductance. And here's an instrument to measure the stomatal conductance. And it's a very simple method to use. We're just measuring the humidity at 2 points along a diffusional path from the leaf out to the atmosphere. So the water vapor just passively leaves that leaf. It travels through a little cylinder pathway and then through a Teflon filter that's shown there. And that Teflon filter has no resistance to vapor flow. If we measure the humidity and the temperature and the humidity at 2 points along the leaf, leaf temperature, we can rearrange all those measurements to measure RVS, or 1 over RVS, which would be the stomatal conductance. If you want a more complete explanation of this, actually, these equations are kind of fun to work with. You can just go ahead and look in the manual for this instrument on Decagon's website. Go through and look at how this is done. But a porometer can be useful for measuring the maximum daytime conductance. Cultivars with high conductance during the day will likely have a low transpirational efficiency, or you can measure at night. And some recent studies have really brought out some interesting things that I think needs more work done on it. But measuring dark conductance, what do I mean? Meaning the stomatal conductance of plants in the dark. Water loss during the dark obviously doesn't contribute to dry matter production or photosynthesis, but low dark conductance actually has been found in soybeans to correlate with low concentrations of CO2 inside the leaf. And those low CO2 concentrations, as you know, that was one of the ways that we could increase the radiation use efficiency and water use efficiency, was to make that concentration in the air and concentration in the leaf further apart. In conclusion, the resource capture paradigm can be useful for identifying and removing limitations to production when water and nutrients are plentiful and light is the limiting resource. We can use this very actually simplistic model to understand our biomass production. Now, it's analyzed in terms of 3 factors that we discussed. The available solar radiation, talked about how changes in solar radiation will affect the radiation use efficiency. Interception by the crop canopy, we talked about how we can go out and actually measure that with a few different instruments. And then the efficiency of converting that radiation to actual biomass, which is that radiation use efficiency value that we have to go and measure. The things that we can actually work on if we were going to try to improve plants and improve them in some analysis of improving the genetics, well, we'd have to work on either their fractional interception or the radiation use efficiency. We know that when water is limiting, biomass production can be predicted by this transpirational term, or the transpiration efficiency, and a knowledge of the water and the amount of water that is transpired. And finally, we know that transpirational efficiency is not constant. We've actually got to do more measurements out in the field to use that model, but it decreases as that vapor deficit in the air increases. And even through the day, that's not consistent. I showed how that worked through that relationship in rice. We also talked about the fact that transpirational efficiency is subject to genetic manipulation. There's another area where we can work on to actually improve the internal CO2 concentration internal to the leaf and actually produce a wider concentration difference, and therefore more photosynthesis and more biomass production. And when we need to make these measurements, when we go out in the field to actually identify radiation use and water use efficiency, use of the proper instrumentation is critical to actually get a value for these measurements and for this modeling. Now, a lot of this information that I've presented today can be accessed on our website on decagon.com. So I'd like you to feel free to come visit us there and access this information. And finally, I talked about a lot of different papers here. There's a lot of background reading. We couldn't cover a lot of this stuff in depth. Maybe that's for another seminar today, but here are a few of the references that I went through, and more on the next slide, that at your convenience you can go through, look at these papers, and learn a little bit more about the things that I was talking about. I'd like to thank you for attending today. I hope we were able to give you some new and interesting information that you can use in your research, and I look forward to hearing and learning a little bit more about what you're doing. Thanks.